Good afternoon, everyone. I'm Rochna Roshan, your webinar coordinator from Center of Excellence for Watershed Management. Welcome to today's session on interactions and connections between soil organic matter dynamics and microbial and vegetal biodiversity. Before we begin, I kindly request all the participants to refrain from interrupting during the session and mute your microphones. Please feel free to ask questions only in the chat box, which will be addressed at the end. Firstly, I would like to invite Dr. Nagaraja N., a consultant, Center of Excellence in Watershed Management, to welcome the speaker and the participants. Over to you, sir, please.
Good afternoon, everyone. I am Nagaraj. On behalf of the University of Agricultural Sciences, Bangalore, Watershed Development Department, Government of Karnataka, Department of Land Resources, Government of India, New Delhi, the WorldBank, and the Special Officer of the Center of Excellence and Watershed Management, I earnestly welcome Professor Samuel Abiven, Professor in Biogeochemistry at the Ecole Normale Supérieure, Paris. It is popularly called as ENS, and also he is the Director of Research Station of Ecology and Geosciences, Paris, France. Now, on behalf of the University, I welcome the Vice Chancellor, the officers and faculty of the University of Agricultural Sciences, Bangalore, all the foreign universities in Karnataka and in India. I also welcome the Commissioner and the officers of Watershed Development Department, Government of Karnataka to this webinar, sir. I welcome the task team leader, Ms. Preeti Kumar and her team from the WorldBank to join this webinar. And I also welcome the Secretary and the officers of the Department of Land Resources, Government of India, New Delhi, and also National Rainfed Area Authority, New Delhi. I also welcome the scientists of Indian Council of Agriculture Research Institutes and other research organizations located in India and elsewhere. I'll welcome all the officers of all the development departments in Karnataka as well as in India, and all those who are registered for the webinar and also who are currently in front of the laptops or in front of the computers participating in this webinar. Sir, again, I welcome everyone for this webinar. Now, I request.
Thank you, sir. Thank you for the insightful welcome address. Next, I would like to invite Dr. N.B. Prakash, our Special Officer, Center of Excellence for Watershed Management, to introduce today's webinar speaker. Over to you, sir, please.
Yes, it is my pleasure to introduce our distinguished speaker of today's webinar, Professor Dr. Samuel Abiven, a distinguished researcher and expert in terrestrial biogeochemistry. I know him very well since 1 decade ago. Born in France, Professor Abiven is a French national and also holds overseas citizen of Indian status. That's why he has visited US Bangalore a couple of times and sent several of his students to US Bangalore too. Professor Abiven is currently a professor of biogeochemistry at the prestigious Ecole Normale Supérieure, Paris, and director of research station of ecology and geosciences at Paris. He is also involved in the Indo-French cell for water science in Bangalore, that is at IASC. Professor Abiven's academic journey began with a master's degree in agronomy at Paris, followed by a master's in biogeosciences and earned his PhD in environmental science and agronomy from France during 2005, focusing on organic matter interactions with soil systems, particularly in carbon sequestration and climate change contexts. Throughout his illustrious career, Professor Abiven has had esteemed positions, including research group leader and lecturer at the University of Zurich, Switzerland, and postdoc researchers at Agronomic Rennes, France. And his research expertise encompasses terrestrial biogeochemistry, carbon nitrogen dynamics, organic matter persistence, and ecosystem resilience. Specifically, Professor Abiven specializes in dynamics of organic matter, biochar, and broader geochemical cycles of carbon. His work delves into organic matter stabilization and decomposition in soils, examining long-term impacts of carbon storage and soil health. Notably, he has advanced our understanding of pyrogenic carbon, which is a new era for work which is happening in the carbon, in soil ecosystems and how land use practices affect carbon fluxes. So Professor Abiven has led 24 funded research projects, published 140 peer-reviewed articles, collaborated with 25 countries in the whole world, supervised 11 PhD students and 7 postdoc students, researchers, and he has as associate editor of soil research and reviews for over 25 top-tier journals, including Science, Nature, Global Change Biology, and he has evaluated research proposals for various national and international funding agencies and organized conferences, including the French Lysimeter Network and deep soil warming workshops. An active member of the prominent scientific organizations such as European Geosciences Union and the International Biochar Initiative. Abiven has been involved in large-scale research projects. So please join me in warmly welcoming Professor Samuel Abiven, a leading expert in the field, specifically on carbon dynamics, in today's webinar, and to share his valuable insights with us today. Professor Samuel Abiven, we are happy to receive once again from on behalf of all the participants today, and also from the University of Agricultural Sciences and Center of Excellence on Watershed Management, and request you to deliver the talk. And those next to Rachana, please.
Thank you for an inspiring introduction, Prakash sir. Professor Dr. Samuel Abiven sir will now present on interactions and connections between soil organic matter dynamics and microbial and vegetal biodiversity. Dear participants, please feel free to ask questions in the chat box, which will be addressed at the end of the session. Over to you, Dr. Samuel sir, please.
Namaskara, everyone. Thank you very much for your invitation. Thanks a lot to Professor Prakash for this great presentation. So I'm going to share my screen. Tell me if it works. Oh, you got my slides?
Yes, visible.
Okay. So yeah, thank you very much for this presentation, for presenting me, and especially for inviting me. So the topic is extremely broad, so I will not be able to cover all of it, but I will give you some glimpse and some ideas about what the topic is about. So as Professor Prakash said, my specialty is more on the soil organic matter dynamics. I do a little bit of biodiversity, but it's a bit on the side, and you will see that I try to integrate it, but please be aware that it's not exactly my specialty. So when we talk about soil organic matter, we talk about like a key component of what can happen in a soil, and mainly something that would determine most of the function of a soil. I took an old paper to illustrate this because this is already known for a long time. Soil organic matter will be connected to biological function, like providing energy, nutrients to the plant and the microbes. It will have physical functions to improve the structure of the soil, the retention of water, and different thermal aspects, and also chemical functions, contributing to cation exchange capacity, pH, and managing to complex cations that would provide elements to the plant, but also reduce toxic and promote organic matter binding to the soil. So actually, all these functions are mostly related to how carbon cycle in the terrestrial system. So this is an overview of this terrestrial carbon cycle, where actually, if I start from the atmosphere, the plant will absorb, assimilate CO2 during photosynthesis, will allocate it in the plant, so in the leaf stems, in the roots, also in the rhizosphere. This carbon will be then deposited, so either during the life of the plant through the roots, so there will be all the rhizodeposition aspects, but also when the plant dies, then organic matter will fall, so the roots will become available for degradation, as well as the stem and the leaves. And this in the soil, you will have different concepts or different processes that can take place. Decomposition, which is mainly the reorganization of organic matter into other type of organic matter. the respiration, so that the mineralization, that microbes use carbon to produce energy, and that goes back to the atmosphere. Relocation, that's typically because of erosion and soluble organic matter going down to the deep soil with the water. And finally, stabilization, which is a key aspect of what I'm doing. So basically, we know now that the carbon stays in soil longer than it should. I'm going to talk about it a bit later. And it's what we call stabilization. So all this is connected to other cycle, like water cycle, nutrient cycle, that comes in the plants and the soil because of because of basically that's the nutrition of the different living elements, but we'll also go out of the cycle when all this degrades. It's very important to remember that this cycle actually today is integrated in conditions that are changing. So global change altering this cycle in different ways, altering the cycle by the distribution, how the CO2 is concentrated into the atmosphere. That will be trends on the long term, that you have dynamics around the season, but basically the dynamics along the long term. Changes in temperature, where you're going to get the temperature varying locally and regionally, but basically with trends over the time. Some extreme events that will strike and then modify very rapidly and strongly this global cycle, and also aspects that are not directly related to the climate, but more to type of pollutions or changes in cycles. And this is the case, for example, for nitrogen deposition coming from industry and agriculture that's falling back on the land. So me, in my research team, what we're interested in is two main questions. How much carbon is there in the system and where it is depending on the conditions, and how fast it goes from all the different compartments during the life of a plant, and after that in the soil, how long it will take to degrade or to stabilize. So some ideas and some quantitative ideas about what we can say about that. When we talk about stocks, actually what we see when we look at the distribution of the stocks of carbon, this is a top left map here, we see that the organic matter is mainly located actually in high latitudes, where you have this permafrost, where you have actually organic soil that don't decompose so much. But actually you still have also places with quite a lot of carbon that are not under this kind of latitude. Sorry? Okay. So you can hear me, yeah?
Yes, sir. Yes, sir.
Yes, please.
Okay, sorry. Yeah. So basically you have this kind of high stocks and you have high fluxes, and not surprisingly, the high fluxes are in places where the conditions are like favorable for degradation. So when it's hot, when it's humid, and you will recognize actually that in between the tropics, you will have this high respiration fluxes that are important. When you look at that in terms of distribution between the soil and the plants, so this is this figure on the top right, you will see that basically the organic matter is mainly in the soil by far, a lot in the top and down soil, and the deeper soil in the high latitude. And when you look at the tropics, you will see that actually we have a lot in the biomass, but also a lot going on in the deep soil organic carbon. So this is how much it is and how it is distributed. How old is a kind of key question, because how old is very surprising compared to what we know about this organic matter. And that comes to this first sentence that we published a couple of years ago. The question is why when organic matter is thermodynamically unstable, does it persist in soil sometime for thousands of years? If you leave a salad in your outside a refrigerator, you know it will degrade very quickly. You know that it will disappear very quickly. So then the question is why in soil we got this carbon that will stay for very long. So this is actually a very nice synthesis coming from tropical grassland and forest. Here you got the depths on the y-axis, and on the x-axis you have a distribution of carbon age. And you see that basically at all depths you will find very old carbon, especially when you go down to the soil where about 40, let's say 40% is about 50% is above 1,000 years. So this is typically organic matter that must have some specific properties. I just give a word of something that is could be taking a lot of time. There will be charcoal, for example, like what you see on this picture, that can actually stay for a very long time in the soil. The way we explain now this persistence to the carbon in the soil is actually that we change a bit the process of the concept explaining this. In the past, we were explaining this persistence mainly related to the chemical persistence. That's probably something that you all heard about. Like you will turn carbon into some very stable molecular structures called humic substances. And these humic substances are coming from the condensation of, for example, fresh litter. So organic matter that will turn into litter and condense, creating these tables that are very persistent over time. We believe now that this kind of chemical persistence does not really exist anymore. So actually that probably these humic substances are mainly related to methodologies that were leading to it rather than observation we can do in nature. We turn now to something that we call ecosystem persistence that are actually more related to the condition in which organic matter are based. If you get different sets of fresh organic matter, you will see that actually, for example, the rhizosphere inputs are probably more important than the fresh inputs. You will see that actually the microbial products actually are probably more persistent just because the way they are built up than the organic matter, fresh leaf organic matter that's falling on the floor. Like that, we identify several aspects, like for example, physical disconnection, the fact that enzymes cannot progress in the soil when they are searching for organic, well, when they are on the way to organic matter, the question of sorption-desorption, the association with mineral association and the desorption could play a role, and so the freeze-thawing aspect. So we left this chemical approach and we went more to this ecosystem approach. And that will lead me very naturally to the biodiversity aspect. So when I want to study decomposition, how do I do? Actually, most of the time, that's my figure from the right here, I will follow the product of the decomposition, product of mineralization over time, meaning the CO2, the methane, the DOC, that's the main one. And I will do that over the course of a decomposition. As you can see here on the left, on the figure, you will go for something fresh that will degrade over time. And what will be left here will be what we call SoilOrganicCarbon, stabilized SoilOrganicCarbon. All this is very classically defined by an equation that looks like this. The percentage of carbon that we lose over time will be a function of the organic matter quality, so the characteristics of this organic matter, by the microbial activity and how this will change over time, by something that we don't really know yet. I think there's still a lot of unknown about this. And this is where basically how we connect to biodiversity. When you look at this cycle, actually there's two places where biodiversity plays a big role. First one is on the plant biodiversity aspect. When you modify the diversity, you will modify the global cycle. And on the side of microbial biodiversity, where in the soil they can act during the process of decomposition and stabilization. So this is basically how these two connects. Maybe I just want to say a word about what means biodiversity, because actually there's three big definitions for it. There's more than that, but there's three definitions. The first one, so main definitions. First one is species diversity, so that the variety of different species within a habitat or an ecosystem. That's really the number of species that you're going to get in one place. The genetic diversity is a variation of genes within species, including differences between population or individuals. That will be really the pool of microbes that you can get, the genes that you can get and that are available in the living biomass. And you got what we call the ecosystem diversity. That would be the diversity of ecosystem, including different habitats, biotic communities, and ecological processes. So actually at another scale, basically, than the previous one. So when people talk about plant biodiversity, very often they talk about that. Actually, I talk about this. So the big difference between the two is that I will not consider diversity of a plant ecosystem or plant habitat or structure, community, related to only the above ground biomass, but only about, let's say, the number of flowers they do, to make it simple, but also about how they connect to each other within an ecosystem, and especially how they connect in terms of rooting. I think the roots have been completely understudied until like 10, 15 years ago. And we still know very little about what's going on below this, below the soil surface. And as you can see on this picture, you have both diversity of biomass, of depths, of organization, of architecture, and all this will play a critical role. So one of the first things that you can see in terms of biodiversity actually is the quality differences that you can imagine within even the same plant between the above ground biomass and the below ground biomass. And I took this example from a previous study of mine a couple of years ago, where we looked at the decomposition rate over the time of soya residues, so root, stem, and leaves. So here is the time on the x-axis. On the y-axis is the amount of carbon that is degraded when you incubate such kind of material. And actually, you see that the root is decomposing much less than the stem and the leaf. This you can explain very well because the root is made of a lot of lignin or components that look like lignin, a lot of cellulose, when you have in the leaf, and especially in the leaf, a lot of soluble components that can degrade very quickly. And you see, like within 10 days, you can get 50% of the material decomposed in a control condition experiments. So this is actually very reproducible result. Most of the time, roots decompose twice less or even more than above ground biomass. And so they will actually constitute very rapidly the organic matter of the soil. So actually, this is now something that is quite well demonstrated, that the soil organic matter is mostly a root-derived soil organic matter. Root is a key component in this. So when we want to study the importance of plants biodiversity and how that promote carbon and inputs and storage, it's quite complicated because if you take natural ecosystems, then you will have no choice. You have to deal with the biodiversity that is on site. One way is to basically manipulate biodiversity. So meaning that you're going to make field trial with one species, two species that you will introduce yourself, 5, etc., etc. And this is actually, for me, the most obvious demonstration about how carbon dynamics evolve with the plant biodiversity. There's 2 big experiments going on in the world about that. Both of them are in grassland, and one is in Europe, and the other one is in the US. I will show you 2 examples, these 2 as examples, and that give also a bit of an idea about the limits. So the first one is the Jena experiment. This is in Germany. And as you see on this plot there, they actually have very small fields where they have either one plant species, either 2, 4, 8, 16, or 60 plants. Okay, so within this plot. And basically, they are actively keeping this biodiversity over the years. So this is no more than 20 years. And what they observed after something like 15 years or 13 years was that if you have globally an increase of soil carbon over the year when you have more plant species, okay? So globally, you have a trend saying that more species you have, higher the amount of carbon you get. what is very surprising there is that, so first of all, the changes are relatively important, but they are not gigantic. So that's also not a huge effect. And actually, when you look at this, and I'm sorry, this is a bit of a complex figure. Actually, what's happening is that when you compare the amount of new carbon that came in, but how much carbon you lost, the all carbon you lost on the way, actually you realize that if you increase the diversity, you increase the inputs. That's a bit what we see on the right, but actually you also increase the losses. So basically increasing the biodiversity can have a kind of dual effect in this case. And this is something that we see in several studies. You increase the new, but you tend to replace the new by old one, and the old one goes away. So that's something that is quite interesting. If we now look at the second site, where they do such kind of experiment at the Cedar Creek Ecosystem Science Reserve in Minnesota, you see with the same time of experiment, you see also an increase of the soil carbon storage rate with the number of species. So this is what you have here, 1 to 16, and the carbon is increasing. But actually what is interesting is that most of it can be explained by root-derived carbon. So actually that the roots will be the one bringing the carbon and increasing the carbon in the soil, and let's say taking advantage of this biodiversity aspects. So that was just a glimpse for the plant. We can come back on that later if you have questions, but basically the trend is that you gain more, but also lose more within high species richness. The root carbon seems to be of major importance. Most probably is very ecosystem specific, and you saw that I show you 2 temperate experiments, and I think we desperately need more tropical studies. So there's few in forest, but that's definitely something we need. And we have many observational studies, but few manipulative. And I really believe that we need manipulative to really understand what's going on there. This is for the plants. For the microbes, the microbes is difficult to manipulate. And you see, I give you a kind of overview of all type of diversity you can get in a soil. I will not name all of them, but basically you get bacteria, you get fungi, nematodes, etc., etc. So you have this large diversity, and this is actually almost impossible to manipulate. So you have to go by big trends over the global, so over the globe. And actually the trends are actually relatively surprising. So if you look at this figure, so I'm sorry, these figures are a bit dense and complex because I try to integrate a lot of things. But if you look at here, the altitude, again, the taxonomic diversity of bacteria and fungi, or the functional diversity of those two, what you realize actually is that globally, it seems that in tropical zones, you get actually less bacterial functionality and taxonomic diversity as compared to what you will get in temperate zone. While in fungi, it's a bit the opposite. So basically, you will get kind of more diversity, larger groups of agent in fungi in the tropical zone as compared to the others. So that seems actually quite counterintuitive. You can imagine that you can rather much more diversity when you have also more diversity in the plant. Well, it seems that it's not exactly the case right now. And actually, it seems that it's quite constant over the places. So when you compare that to the carbon content, so this is what you get here. So on the x-axis, you always get soil carbon content. And on the y-axis, you got different aspects. So here you got bacterial biomass, bacterial richness, that's diversity of bacteria, fungal richness, fungal biodiversity, fungal richness. First of all, you see that carbon content in the soil drive most of this relationship, especially when you talk about biomass, but it's much less the case when you talk about richness. It seems actually that richness is not directly related to the amount of carbon and to the amount of biomass, which is also quite surprising, something that you will probably not expect. So what to say from that? To say that actually we got this diversity and probably capacity to replace each other depending on the conditions, so multiplication of functions, etc. But this diversity is actually quite triggered by the condition around. And this is quite obvious. When it's hot, decompose more, but actually it goes a bit further than that, and especially in the frame of the global change. In the frame of the global change, you see that actually the microbial biodiversity is actually strongly affected by most of the drivers that we observe right now in the question of global change. And I would just cite a few. We know that the increase of CO2 might activate microbes. Increase of temperature will activate them too. The fact that the permafrost is thawing will increase their activity, and in particular producing CO2 and methane, etc., etc. So basically, all this will definitely activate them. And when you look at which are the important parameters that will be structuring the soil community, so especially in this case, bacteria community, you again find organic carbon quality and quantity. They are at the same level in this review there from the pH and the oxygen redox status. So really, we see that this link is actually very strong, and they are both really directly connected to each other. So when a way to go around that would be my last slide or last input there, would be that when you want to look at what can predict bacterial biomass, bacterial richness, that is in this figure here, and the same for fungal biomass and fungal richness, and you do this mathematical calculations to connect them and see what connects to what. Each time, whatever you do, you will end up having soil carbon being the predictor for all of them. So even if they don't connect one-to-one, there is definitely a connection that makes them completely interactive and making basically soil carbon one of the best predictor of all this biodiversity you can get in soil. So just to conclude, soil carbon drive plants are microbial diversity and vice versa. They are really connected. Still many unknowns. I will just finish on that in a second. And most of the connections are ecosystem related. So we really need more of this kind of experimental setup a bit all over the globe to better connect all this. And I just want to conclude with a striking figure that I found in a paper while preparing this presentation. This is a figure connecting the soil functions of these colors that you get there with a different kingdom of life. So bacteria, archaea, protist, plant, fungi, and animals that you're going to get here. And actually, orange is the one that is related to, so that we can relate one kingdom or one tree of life branch to a function. Carbon mineralization is somewhat related to all of them, but the gray is actually for unknown and absent. So what I take from this figure actually is to say that actually we know almost nothing from all this relationship between diversity and dynamics of the soil functions. And with this, I thank you very much for your attention.
Thank you, Professor Dr. Samuel Abiven, sir, for illuminating the integrate relationships between soil organic matter dynamics, microbial biodiversity, and vegetal diversity. Your insights have broadened our understanding of these vital connections. Now I request Dr. Virupaksha to facilitate question and answer session, addressing questions from the chat box. Please proceed, sir.
Thank you, Ruchina. So this was a very interesting topic, and I think it was more insightful. So there are several questions in the chat box. Some have been to everyone and some have been directly to the host. So I have summarized few questions. Okay. So one of them is asking, is chemical persistence degrade faster than ecological persistence of organic matter?
Chemical persistent versus ecological. That's a very good question. I never thought about that this way. So the chemical persistence, is probably actually globally quite overrated because it's probably mostly related to errors that we did in the lab. So yeah, if you spend some time in the lab, you know that to extract these humic substances, we're using strong acids or strong bases. And we think now that these very strong structures that they are, so chemical structures they found out of that is actually probably a bit of an artifact. However, this is clear that you still have molecules that are staying longer. You know, for example, if you take, I would take lipids, for example, they have a longer turnover time. Ecosystem properties, in theory, have a much longer persistence. If I take the example of the permafrost, if you don't touch the permafrost, the carbon there for thousands of years.
There was another question. New carbon stocks and old carbon stocks, is there any balance between them, or is there any trend from last few years?
So it will depend a lot on the ecosystems, clearly. So you get places where you have very old stocks and old organic matter, and losing it is a catastrophe because actually it takes ages to fill up the stock. You have ecosystem where you don't care that much, you know, so that basically the inputs of organic matter are so large then it doesn't really matter. That would be, for example, savanna, so wet savannas. You don't really care if you have old stock, but there's so much input that can come in during the year that most probably you will not have big issues with that. What I would say. But in general, I would say that if you reduce the average age of the organic matter so that you lose all carbon, it's rather bad news because you will need to have even more new one to compensate for it. So it's somehow for me a bit safer to keep old one well stored in the soil.
And one more question. So does anthropogenic activities, especially in African and sub-African countries, imbalance carbon ecosystem? So can you suggest any key measures, some key measures to achieve deterioration neutrality?
So I'm not a very big specialist of Africa. I'm not sure I'm a specialist of any place in the world, but globally, this is clear that we have a decrease of soil organic matter more or less everywhere on Earth. Okay, there's no big places where we can say that the system is still really in balance. For different reasons each time, but you have a global decrease. So how to keep it high or increase it? Well, you have different solutions, of course. You have something that is very drastic and like people propose to put forest everywhere. I don't think it's a very good idea. You have a set of solutions that's called a nature-based solution. So actually solutions that are based from natural observation and that people can implement. So most probably this solution will not bring the carbon very quickly to where it was before industrialization, but at least it goes in this trend. It's very important actually to realize that most of these very drastic actions probably will have counterparts that we don't really know. We're not very good at studying counterparts, at balancing the, let's say, the positive aspect and negative aspect. When we see something positive, we all jump very quickly into conclusion. That's probably a mistake. Yeah. So I would recommend nature-based solution in general.
Thank you, sir. So that summarizes some of the.
Few more questions.
There are some few more questions are coming out now. So, what is the role of invasive plant species in carbon increase in soil and atmosphere?
I cannot say a global picture. So most of the time, these invasive plants tend to destabilize the ecosystems, and then destabilize ecosystem makes them a bit less resilient to global change or impact from other from the outside. So this would be globally rather on the long term a decrease. On the short term, you have some very big surprises. So, for example, in places where I work in India, so Kabini River watershed, for example, we are invested with Lantania plants. And actually, on the short terms, you see that these plants can bring a lot of biomass, but they tend to destroy the global ecosystem. So short term may be positive, but on the long term, I think pretty negative. You can detect them, you can see that they bring carbon and they bring inputs, but if it at the cost of the rest of the ecosystem, this is an issue.
And one more question is there a way we can interlink water credits and carbon credits?
Oula. That's a tough question. That's a tough question
Yes,
because it's very, yeah, it's very regional and very, you know, what I know from the carbon credit situation in south of India is quite different from other places in the world. Can we interlink them? Let's say that on the science base, yes, we can try to make connections, but in a way, water credits are, well, how can I say that? Yeah, well, I think we can try to do that, but I think it's already hard enough to make carbon credits and water credits separate in a political way or in a, let's say, an organizational way, so that we try to even couple them that seems to be quite a challenge. But scientifically, I think we should actually. We should talk much more about water when we talk about carbon.
There are many number of questions, and actually in the webinar we do not allow individual one to ask you because of time constraints.
Okay.
So there are many still available in the chat box. We would like to share it with you. Maybe when there is time available, you can independently answer them also, so that you will have more time, free time to think over and then get the good answer for the participants, Dr. Samuel.
Thank you very much.
Thank you for excellent summing up of the questions and answers, Dr. Samuel, Abiven sir, and Dr. Virupaksha. Finally, I would like to invite Dr. Nagaraja sir to deliver the vote of thanks.
Sir, on behalf of all the stakeholders of the reward program, which is a watershed program implemented by the government of Karnataka, funded by the WorldBank, and the University of Agricultural Sciences, Department of Land Resources, and the WorldBank team, Professor Samuel Abiven for his distinguished presentation on the topic. Sir, we are very much benefited from your mature presentation. I thank you on behalf of all the participants, all those stakeholders who are present for the webinar, sir. Thank you very much, sir. Indeed, it was an excellent presentation. This is the fourth presentation, and it was an excellent one, I would say. And I also thank all the officers who have joined for the webinar, all the participants, all the scientists, the teachers, students, everyone who have participated and got the benefit out of the presentation by Professor Samuel Abiven. Thank you, everyone, and we look forward for your active support for this upcoming webinars also. Thank you, sir.
Thank you, Dr. Nagaraja sir, and thank you everyone for your active participation in today's webinar. Before we conclude, let's take a look at what's on the horizon. With today's webinar, we've completed our in-depth exploration of Theme 1, that is Carbon Connect, focusing on soil organic carbon. Next, we embark on Theme 2, that is Hydrology Unplugged, exploring the fascinating world of hydrology, uncovering its significance. Join us as we venture into this new territory, exploring 4 captivating subtopics each month. We're grateful for your ongoing support and we look forward to your continued participation. Thank you, and we'll see you at our next webinar next month.
Thank you.
Thank you. Thanks, everyone.
Thank you
Thanks,
very much.
Professor Samuel. Yeah, very happy to see you and then listen to your lecture once again. So
Thank you.
I request all the participants to keep supporting and participating in the webinar series on the science-based ones. Next one will be on hydrology. Keep supporting, keep participating in the event. Thank you. Thanks everyone once again.